Greetings and salutations,
Hello, hello.
Earthlings. Hello,
How are
Tim
you?
Ryan. How's it going?
I'm digging the tweener stage do.
Oh, Lord. Why you gotta, like, I mean, it's obvious already I look like a nincompoop, and then now you gotta tell the world. I put a little hair gel in the other day. Didn't like the way it looked. I said I can't compete with Tim today, so I'm just gonna slick it back like Tim. Maybe I'll grow it longer.
I dig. You doing well?
Yeah, I'm great. I'm great.
Yeah.
Nothing to report.
What? I'm sorry?
Nothing to report.
I just have a lot of people who are like, hey, you were able to catch up after Tech Night. Tech Night's it's been 2 weeks, and it's just like, no, I haven't been able to catch up. I'm trying.
What does that even mean?
Or like take a breath. I don't know. I bet, yeah, I mean, there is no time to take breathers.
Shockingly, that was 14 days ago.
That's what I mean. It's crazy. Time
even
just
crazier.
marches on.
What's even crazier is 63 days ago, we had the Cup. And today we have a guest who I believe pitched 9th at the Cup. That's just my memory I'm trying to think. Rachel, you were 9, right?
Yes, I was number 9. Yeah, Yep, number 9. Thanks for having me here.
How did you feel about being 9?
Well, so I see. I typically like going first or second. First or second, but I like the number 9. I see numbers as colors, and number 9 is like fuchsia to me, and that's a good color. It's very vibrant. So
wow,
I like the association there.
Is that a special trait, or is that unique to you?
No, I think a lot of people have that. Yeah.
Wow.
A lot of people have that. I think there is a name. I think it's synesthesia, but it's a lot of people have this like association of not just colors and numbers, but all sorts of things. Like I've heard some really funny ones. Like the color 4 is orange and also Thanksgiving.
Is that because of the third Thursday?
I don't know, but Thursday is also orange. I think
the fourth Thursday. That's what I meant. Interesting.
Yeah.
So fuchsia is like a purple pink color.
Yeah, like hot pink, super hot pink. And I was like, I'm good with that. That feels vibrant. That feels good.
So fun story is this is how the company order happened. Took all 10. I threw it into a robot and I said scramble these, and it came out with what it did. And I said I'm not messing with this again. And we just went with it. Literally the first time.
It's always a controversy. People are like, oh, I don't want to be this number. Or I don't want to be that number. And that's just.
Well, I remember when we had Leland on, and even like beforehand, there was rumblings about him going last. I'm like, dude, that's the best place to go. Like,
people remember
you thinking you want to go 1 or 2. I like when I'm doing performances, I want to go one or last, first or last. Because
why?
You have all the eyeballs, and then you have the last eyeball.
Yeah.
So either you're like you are the one to set the stage, or to be the one that's the freshest in people's minds. And most people in between aren't going to do something that's memorable enough to be that for people to remember it. So
yeah.
But 1, 2, I've never heard that.
Yeah, I like first or second.
What color is 1 and 2?
1 is white and 2 is blue.
Okay.
And 2 is a lucky number. So
what about 6, 7?
Green and yellow, but like a limey yellow for 7.
Apparently there's a thing going on with the kids with this 6, 7 number.
I just heard this for the first time, and I'm like, I don't. Yeah, I'm gonna have to look into that.
Silly. So I wanted to get that out there. So like.
Oh, my kid is 2, so I have no idea what's happening with
teenagers,
the kids, so to speak.
Got it. Well, we're.
I'm just curious. You're from Charlottesville.
Neil, based in Charlottesville. Neil Piper as well.
So do you guys do you both duke it out at different pitch type events, or how does that play out?
I don't know if duke it out is the right word. I admire Neil so much. I love watching him pitch. Before we formed Avant, my co-founder Rana and I sat in a BioHub event. So Charlottesville has this thing called Seville BioHub, where they used to have a monthly speaker series on like Tuesday nights. And Neil came and told the story of his company. And both Rena and I were just absolutely in awe of their founding story. Both he and his wife are entrepreneurs. We just adore them. So hard to duke it out with somebody you really adore. It's more like an all boats will rise situation. I like to think we just bolster the platform for one another.
That's where my head is, just in terms of like you're building a little bit of density there, and like you both can be the show, and you just build around that, and that's good.
Yeah, there's room for everybody to be great, and we're in such different spaces that we don't really have to duke it out. I think we can both win.
Yeah,
Speaking of
I guess
duking
when
it out,
I was like, oh man, here's Neil again. He's gonna. I want my time, or maybe Neil feels the same way.
Oh no, I was so excited to see him get up there. Yeah.
Speaking of duking it out, we've just learned recently that Neil did not take home the World Cup trophy. Very unfortunate. I don't know how that could happen. So I think we need to, we need to send a letter in with our displeasure.
With our Virginia displeasure.
Yes. You gotta get everyone to sign it. Send it in. And we need to let the folks know out there in SF they made a bad move that day. But still super excited for Neil. He's in good spirits. I sent him a couple of texts, and maybe him taking that L will be good for you, Rachel, to get to that. Maybe you guys do duke it out.
I would imagine a national competition like that is probably really tough, because how do you select? Startup companies are all so different, and not just in the problem that they're solving or the market that they're in, but the stage. Even if you say you're a seed stage company or a Series A stage company, that can vary drastically. Right? Are we talking about a seed-based company that has some revenue already, or one that's like in devices where you still need a lot of development and potentially like FDA approval before you have revenue, but you still have a ton of traction? So I don't know how those competitions are judged, but I would imagine it's very difficult to select. It should almost, I don't know. Someone outside of me should probably do the better job of selecting companies that are on equal footing to compete like that.
And I think that Zach and I, we've done a lot of different events like this leading up to this, and it's really like, to me, it's not. I'm a competitive guy, but in this case, it's not about necessarily winning. It's like, who can you meet that's in the audience that you can establish a relationship with and leverage it from that standpoint? $1 million
even the event that you pitched at, that was in all of our communication. Hopefully that was relayed strongly enough, but like, hey, like, you could win. Yeah, you can go out there, but like, what potentially could this be for you, just being here from an awareness perspective, getting in front of the right people, maybe the next person to invest in your company, to be a customer, to be a teammate, whatever that could be. Use it as that too.
It's an incredible platform, and it's so fun, and you get to connect with other business owners. That's part of it. So you're all kind of going through this arc together, regardless of where you are on that journey. Having people to connect with is huge. So I just reconnected with Neil the other night at an event, and just talked about that exact thing about pitching and the persona of it all, and being able to tell your story really well, which is such a difficult thing to figure out how to do, because your story so well, you can get in the weeds on it. It's cool. I was very honored to be involved with World Cup this year.
Let's get into the business. I know my wife, when she watched you pitch, she was like, that is alarming in terms of like status quo and the problem that you're trying to solve. So yeah, let's get into it. What is the problem that you're trying to solve? What is the business? And then we'll dig deeper from there.
So Avant Genomics was founded by myself and Rena Nawari. We were laboratorians. We worked in a bioanalytical lab doing chemistry work, but also developing lab tools. So everything that a lab tech sees on their benchtop, the instruments, the consumables, putting those things together in a really intelligent way that makes them very usable. And for me in particular, I was focused on the sample preparation portion of that workflow. So taking genetic material, DNA, and pulling it out of something like blood or a swab from a cheek or something like that, and doing it really well. My background is in forensics, where sometimes you just get such a small amount of DNA that you really have to work hard to consume it, and these processes are really detrimental to the integrity of that sample. And after years of working in that space and going to conferences and seeing that other people were having this exact same problem with this very labor-intensive, time-consuming workflow in the lab that was also leading to a bunch of DNA loss, we decided to start this company. We're now developing a benchtop device for DNA sample preparation for blood-based cancer detection, otherwise known as liquid biopsy. This also includes consumables, so basically a plastic cartridge where the sample goes in, and the user can go from dozens and dozens of steps to just placing it on the instrument, hitting a button, and walking away. So that's what we've been working on here at Avant. Why are you laughing?
Because I just feel really, really unintelligent.
Oh no, I don't want to make you feel that way.
No. I think I'm just like, how do you even like? Did you just follow the path of least resistance to get to where you are today? I mean, it's just like, it's pretty cool how you ended up doing what you're doing. You know, it's just like,
I think it's the path of most resistance, actually. It's continuing to ask problems. I mean, this is just, I was listening to your podcast, a few other episodes. It sounds like you guys get, on a personal level of people and their identities. And for myself and for Rena, I think I can speak for her too. We choose the path of most resistance, and that is we have lots of questions about the work that we're doing. It's never just accept what's in front of you. It's sort of well, why do we do it this way? And if we're having all these issues, why haven't we made a change? And innovation is born from that, from asking questions and being stubborn about not accepting kind of a very base answer.
Yeah.
And so, yes, that is it's the path of most resistance, I think.
My thing is really like, when you were growing up in Jersey, there was just like, hey, this is what I'm going to do. When I grow up, this is my path. Yeah. It just, maybe it was. I don't know.
Well, so I was born in Jersey. I didn't grow up in Jersey. I grew up in Denver mostly. Yeah.
You don't strike me as a Jersey girl.
I don't know how to respond to that.
I don't know why I said it. I don't know what it would look like. It's just,
Yeah, I don't know. I don't know what a Jersey girl. I guess more. So my whole family, my husband's family is all from New Jersey and grew up in New Jersey. And I guess if I'm to look to my sister-in-law, who's a Jersey girl, the only difference between I suppose she swears more in traffic. I think that's about the extent of it. Yeah.
So do you think, I guess back to Tim's question, like, do you think that like your childhood led you to this? Like, were you, were you into like crime shows? Were you into things that?
Oh, forensics led me
Yeah.
to forensic DNA. I don't know. No one's ever really asked me that before, if my childhood led me. I mean, I grew up in the 90s, which is the era of, America's Most Wanted on TV at night. And my sister's dad was a police officer. So I did get to come home frequently to stories about crime, but I wouldn't exactly say I anticipated this is what I would do. I've always just followed my curiosity and my stubbornness, and I think that's what led me to doing what what I've selected as a career. And first in forensics, it was definitely curiosity.
It's the first word that comes to mind when I hear forensics crime. It always goes to Dexter. Are you a fan of Dexter?
I am such a fan of Dexter. Yes, it is. It's one of those, and this is kind of funny, but one of those comfort shows I can rewatch over and over. My sister and I both do it every about year or two. I'm like, I think I should rewatch Dexter again.
Is that the serial killer?
Yeah.
Yes, yeah.
Never
but he's
seen
like a serial killer with a conscience that kills.
Yeah,
people.
you root for the bad guy, so to speak.
A bit, yeah. And you don't want to. It feels uncomfortable
Right.
to do so, but you're like, man, I really like this guy.
Good storytelling then.
Yes, very good storytelling.
So then from forensics to what you're doing now with Avant, very different.
You know, at its core, it's not. I mean, there are aspects, certainly. So when I was a forensic scientist, I was working in the crime scene unit for Baltimore City Police for a while. And then I worked at Prince George's County Police in Maryland, doing DNA analysis and serology. And I taught at Towson University. I taught crime scene investigation in their master's forensic science program. A little different than what I do today, but at its core, it's all just it's science, right? It's dealing with difficult samples, looking for genetic material, and what we do today is exactly that. It's very difficult samples, trying to automate these processes and make them easier to interface and standardize them as well. Forensics is big on standardization, making everything flow in such a way that the results that you get are defensible in a court system. And we don't have that exact same kind of oversight in clinical oncology where we are focused now, but I guess you could consider the FDA the equivalent.
In terms of this science, where do you think we are and like in the knowledge scale? Do we know like we still only know like 1 or 2%? In the grand scheme of things? I mean, it seems like we're always discovering new things.
I think there's always a lot more to discover. I will say discovery now, there's a lot less basic science to discover because a lot of the groundwork has been laid. So we have to think outside the box, maybe a little bit more. And this is, definitely my own opinion. But I think there's a lot to discover. There is a lot in liquid biopsy and understanding the genome, and not just the genome, but what we call the epigenome. So all of the chemical modifications that can be made to your DNA that contribute to different outcomes, how we age. You ever see those studies of how twins will age differently based on the life decisions they make? A lot of that has to do with their own epigenetic makeup. And there's still so much to unveil there and understand really well. I think this next 20 years is going to be so exciting for genetics, for science.
DNA is fairly new, or the access to something like I worked in TV from like 07 to 10, and I feel like that is that a true statement? Like or has DNA always been around, but you couldn't track it, you couldn't test it, type of thing? Like, isn't there some sort of statement somewhere around there? Because I remember like there was like a story. What's that parkway by you, Tim? The Yorktown Parkway, the Colonial Parkway. Yeah, so like they had never found anything, and then somehow they had saved some material, and then they were able to test it 30 years later. Like, is that just because stuff has gotten better? Like, what am I losing my mind from a memory?
Oh, so if you're talking about forensics and genetics and being able to identify people, I think the 90s is really when that started taking off. I mean, we
Got it.
all
Okay.
have the infamous, if the glove doesn't fit, you must acquit. The
How
first,
did I
like,
know you were going to
tenet.
say that? How did I
Yeah.
know?
So like that. Well, I mean, it's a one of the first examples in media. And back then we were doing a different kind of DNA analysis called RFLP. This has since changed. Now we're looking at something called short tandem repeats in genetic material and looking at so many of them that your level of confidence increases dramatically, that you can, be confident that you've matched the right person. And match is a terrible phrase. I shouldn't quite use that, because it's really a ratio, likelihood, statistical probability. But yeah, it's gotten easier, and the technology has gotten better, certainly. Yeah.
Wait, so are you saying you could 100% know that the person is it? Like, literally, you could be testing something of mine that I gave you. You could test it, and you couldn't say 100% you know it's me. You have to use those other words that you just used.
Yeah, it's like a 1 in a bajillion chance. Like they'll give you a crazy statistic. It's like a 1 in nonillion chance that it could be someone else in the population.
Got it. That's probably if we're living in a simulation then. Okay, got
it. Yeah, but you're like, there aren't that many people in the population, so
Yeah, like you could literally prick me. Okay, that's crazy.
Mm-hmm. Yeah. So you mentioned in 20 years time, there's a lot that. What does that look like? Everything goes perfect for you in the company. Where are you at in 20 years?
The idea here is to make this kind of testing ubiquitous, so everyone has access to it, as opposed to a select few who can afford it or who know about it. These liquid biopsy tests, as I mentioned, are, some of them are fantastic in terms of sensitivity and specificity, how specific we can get for specific cancers and in specific areas of the genome and the body. But I think once we have a really fantastic platform that labs can utilize as kind of the baseline expectation, there will be the ability to input more liquid biopsy tests in more hospitals and more local laboratories, so that more people have access. That's huge.
I don't know. Wow. I don't know. As fast as everything's going, I guess we have to make sure we do everything correctly and we don't move too fast. I don't know. I'm just.
This is one of those episodes where, like, I'm also too dumb, like Tim, to really know a lot. And I just sit back and watch and listen and say, okay.
Well, I mean, it's just from like the
I guess
AI
those TV shows were wrong.
Well, from the AI standpoint, it's just like, man, we got like 5 good years left. And then after that, man, it's just like, we just sit back and just try to figure out what we're going to do with our life, So it's just like, I don't know. I'm just trying to piece it all together right now.
No, I don't think we're ever going to be there. There's so much more to learn and discover. Not at all. There are so many new therapies that are going to emerge as a result of the incredible work that's being done with CRISPR, for example. I mean, and where liquid biopsy fits into all of that, or blood-based detection of cancer. I mean, we're going to be able to select more intelligent therapies. And by that, I mean, using the genome of your own cancer cells to select which ones are going to be the most effective. That's huge. And then monitor patients throughout their entire journey without doing invasive biopsy the entire time. So just taking a routine blood draw, and then monitoring them post all of that to see whether or not their cancer has reemerged. And before anyone even goes through that process, the idea that we could save so many people by taking a blood draw for early detection. I mean, the criticism of detecting cancer early right now with blood is just that sensitivity is a problem, right? If you only have a teeny amount in your body, how likely is it that you'll be able to pick that up with one of these tests? And the idea with what we're working to do at Avant is make the process, the scientific laboratory process better so that you're picking up more of that. So that all of these people who are working so hard to develop these tests have a better chance of picking it up. So what that can mean is you pick up a cancer years sooner than you might have otherwise from just a blood test before you otherwise would pick it up on a scan. I mean, I was talking to a doc the other day, a precision medicine doc, and he has all of his patients do a GRAIL test, a liquid biopsy test. And he was saying the number of head and neck cancers that he has picked up early from that test is astonishing, when they couldn't pick it up on radiology. So I think that we're just starting to really go in this direction, but there's a lot of promise there. And companies are putting a lot of effort and capital into this right now to try to, make this more equitable for more people to take advantage of. So it's exciting. It's an exciting time, but I think there's a ton to learn. And it's not just DNA. We can start looking at, RNA, proteins, all sorts of other things that we'd find in your blood sample. So there's a lot there. I think we'll go far in 20 years. I think there will still be way more to learn in 20 years. Yeah.
That's cool. I mean, it just makes sense, right? Like I feel like the wearable technology so that we can be getting data on a more frequent basis has become such a norm that something like what you're doing totally makes sense, and figuring out just how to do that on a more quicker basis than having to schedule an appointment, have some sort of feeling, whatever majig.
Mm-hmm.
It just makes sense. And so what? Am I wrong, Tim?
No, I like your technical terms. That's all.
The whatever majig. I mean,
whatever majig. Yeah. No, wearables
I'm
are
just excited
so
we haven't
cool.
had crazy.
Wearables are so cool. Yeah. I wear a wearable right now. I'm wearing an Oura ring. That's a wearable.
Yeah. What's your readiness score today?
Started as the start. Oh, that's a great question. I don't know. I forgot to wear it last night, so I don't know if I'll get a readiness score for
What was yours, Tim?
Yeah. I'm a 89.
89. I don't think I'm ever an 89.
Is that good?
Yes, that's good.
Out of 100, yeah.
Yeah, he's crushing it today.
What's your average?
My activity score was an 80s, a 97. Sleep was only an 80 though, so
lacking.
Yeah, I had it on the charger last night, so I don't have a readiness score today for you.
What about yesterday?
So sorry.
I triple crowned yesterday, which I was super proud of.
You
triple crown. Wow. A little Kentucky Derby action.
You're better than me. My readiness score yesterday was a 76.
Ooh,
it was like, it said you need to pay attention to your sleep.
Yeah, that I struggle with sleep. My activity score.
I had a good sleep last night. 8 hours and 43 minutes. Let's go.
8 hours and 43 minutes. Who has the time?
It used to be a little less, but then there was an acquisition of blackout blinds, and it has really changed everything.
Oh, yeah. Mm-hmm.
Yeah.
Yesterday was also an 87.
You're crushing
87 today and yesterday. I guess I went down too.
Congratulations, sir. What finger do you wear that on? Your ring finger?
I use it as a wedding ring. I know that most they suggest the index finger, but
scientist, I like to follow rules, so right on the index finger.
That's cool. To the layman, is Virginia a super scientific new innovation state?
Yeah, I think we have a lot of incredible academic institutions here that make us that. UVA, VCU, Tech. I mean, yeah, absolutely. There's a lot of really cool innovation, especially med tech. I think also I would throw Maryland in there. And, if I'm to turn the conversation back to liquid biopsy too, I think Johns Hopkins University, Bert Vogelstein, I mean, that's kind of an epicenter for liquid biopsy. So yeah, absolutely. A lot of startups, really incredible technology coming out of the Maryland, DC, Virginia zone for
The
sure.
Mid-Atlantic, I guess. Because I think that's the one thing that I've been most, alarmed isn't the right word, informed about just from the World Cup the last few years is how much med tech there's been. And obviously you're in that. The last two winners were med tech. And I don't know, until last year, and definitely reinforced this year, I really would have never really thought of this region, this state as a med tech place, but I guess I'm being proven wrong. Yeah. I mean, obviously the schools that you just talked about.
Yeah, the hospital systems that are part of that school are very innovative. We have a lot of funding going into those programs for science. I mean, just here, Paul and Diane Manning, I mean, they're champions for med tech. And then now we're getting this new Virginia initiative, the Lab to Launch. Have you heard of this? You heard of the new Lab to Launch initiative?
I saw the headlines, but
saw the headline? Yeah.
That's it.
I mean, just kind of coming into a new era of standardized licensing agreements from universities to startup companies to incentivize people to bring their technology to market, as opposed to just trying to pitch these kind of newer academic technologies to larger companies who'd have to still put in a lot of R&D dollars to get them moving. I think it's going to be huge. I mean, this is still just starting. I think it was announced in September. But a lot of promise there. It's really exciting. I mean, I think we have a lot going on. We have a lot of new incubators popping up everywhere. I mean, we exist in one. Avant works in the Commonwealth Bioaccelerator in Charlottesville. There's we now have a little more lab space in town than we would have otherwise, and I think this is true in Blacksburg as well. So I think we're fostering that kind of innovation. Otherwise, it would be hard for it to exist. But there are places for companies to kind of matriculate into, and there's funding, there's government support for us to start these companies. I mean, think of like Virginia Innovation Partnership Corporation, right? So our first funding was all non-dilutive grant money when we were still grad students from VIPC, Commonwealth Commercialization Fund, the UVA Cancer Center kicked in a few $100,000. The Licensing and Ventures Group too, they're supportive and put in on that first round. So those are all Virginia. That's all Virginia ecosystem. That's just putting us up. The Coulter Foundation at UVA. I mean, yeah, they were the first people to say, yeah, you have a really good idea. Let's give you some money and show us some data. So we've just got a supportive crew, for lack of a better phrase.
And your football team's playing well, so.
Yes, they are. I just moved into a new neighborhood. We have some of the coaches in our neighborhood, and so we're on like text chats. So if I don't know exactly what's going on with the games, I still get like updates, and it seems like we're doing pretty well, I think. Yeah.
What are they ranked? 18?
Top 20. I know that. I think they're 5-1. So, how much peer-reviewed literature do you utilize on a weekly basis?
Do I utilize? Like read?
Yeah. I don't know. It's not a question from me. It's from Colin Smith in the comments.
Oh.
I mean, that's way too technical of a question for me. I'm just rereading it. Like, come on.
No, I read a lot. That's part of the fun and the challenge of having a company like this is staying on top of the literature. So, great team. They send me a lot of articles. I read a lot of them. I just got an iPad so I could sit in bed at night and read articles instead of scrolling. But I'd say, several articles or app notes a week.
That's why your readiness score is so low, because you got the iPad studying late at night.
That's the problem. It's the blue light, and then I can't come down until like midnight. Yeah,
they say that's a real thing. 3 hours, don't have a TV or a screen in your room.
Oh God. See, I just got the blue light glasses. So I'm justifying my reading at night with the blue light glasses.
Wait, so how does this work?
It doesn't. It's just supposed to block out the blue light, so your melatonin is like signaled to, let you. It lets you sleep, I suppose.
Can I ask you like a science-based question from like a rookie? Tim
Maybe
can't
I'll
wait
be
for
able
this.
to answer it. Yeah, sure.
All right, so you got like all these like health and fitness gurus that like come out with all this like nonsense. Like, how much of it do you think? And it's too general of a statement, but like, you hear things like red light therapy, grounding, I don't know, not eating before intermittent fasting, things like this, and there's like some sort of like science behind it, but it's like, is this? I guess my question is like, the things that they're saying that are scientifically backed, are they really? And how do you know to like that it's not propaganda, that you're not just being advertised towards, like, kind of thing?
So personal opinion, most of it's probably really good marketing. Some of it, if I hear the phrase scientifically backed, I want to see a published study that's been peer reviewed, certainly. Like I believe red light therapy, there are some published studies on that, if I'm not mistaken, as being beneficial for, I think, cell turnover, possibly. But everything like that, it's really clever marketing typically, and some of it might be true. I think both things can be true, that you have clever marketing and some scientific backing, maybe.
Sure, but you guys are both wearing an Oura ring, so something clearly in that convinced you guys that it was
the Oura ring for the ladies. The Oura ring I purchased because they partnered with a company called Natural Cycles, which tracks your cycles for you, your hormonal cycles during the month, and that is FDA approved. So that's why I got the Oura ring. Yeah.
There you go.
Yeah. So which is why I don't typically track the readiness score and everything else. It's more of a tracking my hormonal cycles during the month. Mm-hmm.
Yeah, I try not to become, just for lack of better term, a slave to these wearables. I mean, in terms of my mattress, my watch, my ring, and it's just like, I feel like I'm constantly. In some cases, it's a good thing. Like, I need to go to bed. My body, I'm getting the push notifications that it's time.
It's time to wind down. That notification. To wind
down.
I'm usually still working when I get the "it's time to wind down" notification, which is I just kind of.
Is that because you set a time that you want to go to sleep at, and it knows that you haven't, or is it?
No, it finds your body's ideal time based on, I forget what they call it. Your chronogram.
Chronotype something.
Chronotype. Yeah. It's like the ideal time based on your daily body signaling. I don't know. I'm not an expert in that.
But it is interesting in the sense of like when as it monitors your body temperature and it tells you, hey, slow down. We're detecting that you're going to get a cold, or where there's so I do appreciate that.
It is fairly accurate with the cold thing. It'll typically alert me like a day before. Be like, you're probably gonna start feeling terrible tomorrow.
Oh yeah.
See, now I appreciate that. All I needed to do was have you on episode 284 of the show to figure out what a scientist is looking for in some of these ads. So thank
FDA
you.
approvals.
Okay.
284. Wow, I'm honored.
We are honored that you're honored.
Yeah, that is always the neither here nor there. But yeah, so like from the fundraising standpoint, is it becoming easier now that there is a sense of density around Charlottesville, Richmond, Virginia? Is that starting to spread further out where it's becoming easier to raise? Because I think the thing with this, it is such a long timeline, and the roadmap is so long, and not all investors are that patient with their capital.
You're talking about for deep tech, lab tech. There's a long
yeah.
timeline before revenue. Yeah, absolutely. So finding the right investor is huge in a business like ours. I don't know that I'm the right entrepreneur to ask whether or not it has gotten better, because we've been raising. This is our first true raise that's a dilutive round. So, it ebbs and flows. And our first checks in the door for our seed round were not actually from Virginia. They were outside capital. But since then, it has gotten easier to an extent. I mean, people are certainly more aware of Virginia. And I believe through VIPC, there has been funding given to several different VCs with the idea that it should be invested in Virginia, which is fantastic. But like anything, it is difficult, and you have to find the right people, certainly. And some of them are here in Virginia, and some of them are not. But we do have intelligent investors here, absolutely, because a lot of them are coming out of these programs. They're familiar with the biotech from UVA and what generally we're subject matter experts in there, and VCU and Virginia Tech.
Yeah, I don't know. It's just, I work with a lot of deep tech companies, not on the med tech side, and it's just trying to figure it out, crack that nut. And your point is spot on. Just not all investors are the right investors, and you have to find the right ones.
Yeah, I think there's a lot of, it's no secret, there's a lot of AI coming out, and we do have a component of like ML in our system. However, what's great about those companies is you put in some effort, you start to build a model, and then pretty quickly you can start testing it with your betas. And so having that capital materialize into revenue can be fairly quick compared to a business like ours, where we have to go through the entire arc of manufacturing before we can send the device out to betas, and that just takes a little bit longer. So I don't want to say patient capital, but someone who certainly understands that this does take a little bit longer, and the risk factors are different, but the payout can be huge.
Yeah, I don't know. I'm just, after listening to all the different jobs that you've had, along your career path, you're like, let's add startup founder to the list, because I need more of a challenge than what I've already
Path
challenged
of most
myself
resistance. Yes.
Path of most resistance. Well, no. So, the story is actually a little bit more fine-tuned than that, right? I was working in forensics for a really long time. My frustration in forensics, which probably would have been a frustration anywhere, if I'm really being honest with myself, which is that with the one foot in academia, where I was going to conferences and learning about new technology, I wasn't seeing that materialize in the lab very quickly. I wasn't seeing really cool NGS techniques, next generation sequencing, come into the lab and really disrupt. And that's for good reason, right? We need to be measured and slow and validate these things. But I wanted to see these technologies be packaged in such a way that we could take advantage of them in boots on the ground more quickly. And I felt like there was kind of a lag in that connection there. And so I quit my job working in forensics to go back and do my PhD in analytical chemistry in James Lander's lab. And James is known very well for developing automated microfluidic tools, so integrated fluidic systems that can make these workflows more robust and rapid, and so on and so forth. And my first year in that program, I wrote an application to the DOJ to develop a sample preparation technology for forensic genetic identification, and it was funded. And myself and some other members of the lab worked on this for several years. And my co-founder and I were at a conference, and I've told this story a few times, so if anyone has heard this before, bear with me, but we went to a conference in Croatia. It was like a really cool precision medicine genetics conference. So it was like precision medicine and oncology and forensic human identification. So anything cool and cutting edge you could do with DNA. And we were actually there getting an award for what we'd been working on. And we were like astonished that we were there for this award because the people that were there were just so tremendous, incredible, forward-thinking leaders in the field. And this was like a young investigator award. So they were like, you're on the right track. Here's, a plaque and $500. But we sat there in the audience and we were like going at the end of the week. So we had the entire week to listen to everyone else speak and were astonished by this really incredible tech that they were coming out with. But all of them were trying to solve the problem of very small amounts of DNA in their sample on the back end by developing a really cool genetic panel or developing a really cool algorithm that could, search through the noise to find the signal and determine whether or not the result you had was a true result. But few of them were focusing on something that we all kind of take for granted, which is just like preparing the sample and how much you lose during that process. And as the week progressed, Rena and I just kept finding ourselves looking at each other like, well, we're trying to solve that problem, but in a different space. And at the end of the week, we got on stage, we gave our talk, and there was a line of people asking if they could use the tech, buy the tech. And at the time, we're grad students. So we're like, well, we didn't really consider building it for commercialization that way. We kind of published it to share it with the world, but we hadn't thought about it that way. So we that night went to dinner with our husbands, who I always say this, they passenger princessed along with us to this conference, so that they had an excuse to go to Croatia. So they're just frolicking about the whole week. So we all get together and we're like, hey, starting a company is a real thing. We think we want to do this. We think we can bring a lot of value to all of these people who are having this problem. Everybody on board, say aye. And then Rena and I, to consummate our union, jumped into the Adriatic Sea together.
Wow.
Yeah, we got back to the US, we founded the company and started going after funding like that month. Tested it with the UVA Cancer Center to really kind of de-risk it for ourselves and for this new application, because it wasn't, it was a forensic technology. So we had a hunch that it would work for cancer detection, but that has to be validated for us and for investors. So we spent a year doing that. Data was good. We had really great concordance with the UVA Cancer Center pathology lab. And here we are today, giving demos in our office.
Silly question.
Yeah.
How often do you hear the word Theranos when you're out there talking to people?
Well, I'm a woman, so probably more than most. But I'll tell you what, I can never wear a black turtleneck again. And it's a shame because I look really cute in a black turtleneck. No, we hear it sometimes. I mean, we're doing something very different. We're using, First of all, we're focusing on one thing. We're also focusing on a large volume of blood, which is why I hesitate to call our platform a microfluidic platform. It's an integrated fluidic platform. So we have a large volume of blood going in, the typical amount you would have from a liquid biopsy blood draw. And that slowly reduces that volume as you move through the reaction so that, the physics of it make the output a little bit better. But yeah, we do hear it sometimes, but we're not doing, 200 tests in a box or what have you.
Well, and it's so silly. It was such a media frenzy that it was so easy for them to jump on board and that people just were glued to it. And it's just like the whole thing was just super unsophisticated investors that had no idea what they were doing. And they're throwing a ton of money at something. And there was no milestone based anything associated with it. And it was just, I don't know, the whole thing obviously was very unfortunate, but.
Yeah, I was not in the inn during that time. Was still in my forensic days, not my microfluidics days. But it's certainly talked about at microfluidics conferences still.
Wow.
And that's the criticism of microfluidics, is that you have to make sure that the targets that you're going after are amenable to low volume integration. So microfluidics is typically based on super, super low volumes of things. And when we're talking about detecting any analyte, DNA, RNA, something that's really, really small in comparison to the large volume it's in, if you're only taking one small representative sample from that and running it through your prep process, the likelihood that you're going to get a good result is low. And so not everything is eligible for microfluidic integration, including, liquid biopsy in this case, which is why you have to start with a large volume of fluid. And you can enrich it down and do a lot of things to make that workflow amenable, but you just have to be knowledgeable about how you approach the situation. And I think some of that nuance was lost depending upon which test we're talking about. Yeah.
Idiot of the Year award. What makes cancer so difficult to find early? And like, why hasn't there been a cure type of thing? Like, it's just like, it seems like the one that like we don't have a cure for, yet it's the one that all the money goes towards. Like, what we seem to cure a lot of things. Why can't we cure this one? Like, what's so difficult about it? Again, remember, Idiot of the Year.
Yeah, I don't know that I can speak to the rationale behind not having a cure. I'm not an oncologist by training. I can certainly respond to the first part of that question, which is why is it so difficult to detect early? So a lot can be missed. It can start off as very, very small. So if we're talking about a scan, the other element of that is many cancers do not have a method for surveillance or early detection. If we're talking about blood-based early detection, this is often based in, circulating tumor DNA. So that tumor has to be able to shed some of that DNA into the bloodstream. And you really only get a snapshot of what is in the bloodstream for a couple of hours because it doesn't last very long there. So being intelligent about how you parse apart that signature is really important, and the approach that you take. And again, these methods aren't available everywhere right now. So, it's a culmination of factors. But if we're talking about, and we bring this example up a lot, if we're talking about colorectal cancer as an example, we have an excellent screening method, right? We have colonoscopy, and it is the standard of care, and it's fantastic because you can visualize polyps. But of this recommended screening age, starting at 45 years old, 50 million people a year in that screening age range who are recommended to get colonoscopy don't get it. And so you have to ask yourself why, because many of them will test positive and it could have been intercepted. So very frequently, this is a lack of access to a screening method like this. So it could be a cost barrier, but it's also, we frankly don't have enough gastroenterologists to provide this service. I think when my own husband went to go get his colonoscopy early, because it runs in his family, I believe he waited 9 months to get that appointment. And when he did get it, there were 5 precancerous polyps. So, it's tough. It's really tough. It's also having people comply with the recommendations.
Okay, good to know. What were those age ranges?
45 is the recommended start of screening for colonoscopy.
3.5 years to go. All right.
You got it. Unless it's in your family, unless you have a family history, then you can get insurance to cover it sooner. And it's worth it. Do it. Go get colonoscopies, everyone, and blood draws.
What's something? See, he doesn't even know he's on mute. He gets so mad at this too. It's so funny. It brings me so much joy. He has planes flying over his house right now because he lives in a flight path of F-18s, I think.
Mm.
And
I was going to ask, what advice do you give? I'm sure that you interact with students. You're in a college town. When students come to you and ask you, "Hey, I'm thinking about, I want to get involved in med tech, thinking
Mhm.
about taking the leap and going and doing it." What's your advice to them?
My advice to them is typically get out there and explore your options. Getting involved in the community is huge, and understanding what your path will look like will help with, building expectations. So for Rena and I, before we really truly started the company, we were going to local biotech events. We were going to Virginia Bio, we were going to Seville BioHub, we were going to startup conferences just to watch people pitch. We became student members of our local angel group here, Cav Angels, which allowed us to watch not only entrepreneurs in the seed and pre-seed and Series A stage pitch their companies and talk about what's important to the narrative of their business and how they told that story, but then how the investors thought about the value of what they were doing based on what they presented to them. I think understanding the dynamics of all of that are really helpful. And then ensuring that the technology that you're considering trying to bring to market is really fitting a need is really important. So what I mean by that is do some customer discovery, talk to some folks that you intend to sell this to. It's not just about building something cool, which is very often the motivation in academia. Like, sure, absolutely, we want to help people, but also it's this really cool thing that we can't stop thinking about. And so we want to build it and make it better and faster and so on and so forth. Make sure that the value proposition that you visualize is true in the market. Are other people as interested in spending money on what you believe they will spend money on? That's really important. And once you can kind of de-risk it through that framework and understand what you're in for, then go for it. Be bold, and then give yourself some grace too, because this is all about growing and learning. No one was born an executive or a founder. So humble yourself to feedback and know, this is the hard part, is you get a lot of feedback. Knowing when it's important to take that advice or just kind of take it under advisement is huge. So just approaching it maturely, I think it's the hardest part.
I think that's really good. It's take everything, but then sift through and figure out what you actually should use. Kind of like in baking, right? Like you put something into the colander, you're not going to use, the crap underneath of it. You're going to use the stuff in. So it's called a colander. It's
I'm a terrible baker, but yes, I know. Yes,
well,
taking all the information. Yeah.
Terrible baker. I mean.
Yeah, I think there's that, and then, but then there's also this point at which you can take in too much information.
Absolutely.
So reading too many books about how to be a really good executive or run the perfect lean startup and all of that, I think it's good to always continue exploring all of those theories, but then at some point you just have to do and make the mistakes and be okay with making the mistakes and give yourself grace there. So it's a good balance for sure.
Yeah, that's great.
Baking's kind of like eating, Tim.
Too true. What if Zach and I take a trip to Charlottesville, where's the place we have to go?
For eating or baking?
For a meal, yeah. For
a meal. Okay, so one of my favorite places in town, it's called Bell. It's a little eatery that during the day is like a coffee shop, bakery, sandwiches, light fare. It's in Belmont, which is a really cute, quaint, older neighborhood on the other side of the downtown mall. And they, within the last 2 years or so, they now have an evening dinner service, which is fantastic. and they source a lot of the like local ingredients, and so the menu changes all the time. But it's very cozy, very Charlottesville. I just adore them, and the founders are great too.
Is that the
I'm
thing
not-
about Charlottesville? Is all like farm to table? Is that like what they're known for?
Yeah, that and the sourdough. I don't know if that's kind of
really?
a claim to fame here is our sourdough. Yeah, we've got a lot of incredible bakeries that make fantastic sourdough, and they're, sourdough has like a lore. So different batches are, going back 100 years.
Right.
Someone has had
The
this
starter.
the starter, that's right, has had this like living starter that they've nurtured for so long. And all the bakeries are kind of like, well, our starter comes from here, or our starter comes from here.
Mhm.
And you can tell you can taste the difference. So Bell's sourdough is very like chewy and like really sour and yummy. Yeah. Yeah, I saw that pinky come.
Yeah. I don't think I'm allowed back in Charlottesville after my last trip. So
What did you do?
I can't tell you on camera.
Oh man.
No, this thing's being
Yucking
recorded.
it up on the downtown mall. Okay.
I don't know
Where
what
was
that
the
means.
place? Where were we at?
No.
Miller's. Yeah.
Well, you brought it up. Oh, Miller's. Yeah, I was going to say it's one of like 3 locations where that would happen. Yeah, Miller's. Okay. Yep.
Well, we appreciate your time today. This has been lovely, learning a lot.
Changing the subject.
Yeah.
That was a great
For all of
segue.
humanity, I wish you the
Really great
greatest of
back.
success.
Well, thank you. Thank you. I appreciate it. This was really fun. Having kind of an impromptu conversation with y'all this morning.
Yeah.
Please do come visit Charlottesville. We'll hide you somewhere, Zach.
Will do. All right. Well, we appreciate it. Hope you have a great rest of your day. Thanks for listening.
Thank you so
and,
much.
Continuing success.